about the thalamus with a focus on epilepsy and conclude with deep brain stimulation of the thalamus in patients with epilepsy. The word thalamus is Greek for bedroom and was first used in the 2nd century after Christ by Galen. Of course, he didn't know the role of the paraventricular nuclei in the sleep-wake cycle, so Galen, Willis, and later the French Louis used the word thalamus because of the morphology. The thalamus has a very close relationship to another diencephalic structure, the optic nerve, and Louis also referred to the thalamus as the couche optique, the bed the optic nerve was resting on. All the incoming information of the body and brainstem has to pass through the thalamus before going to the neocortex, and all the neocortical information has to go through the thalamus first. And you can see on this macroscopic specimen how the thalamus is sitting on top of the brainstem with a very close relationship to the caudate nuclei. As written on the Brain Bit by Bit website, the thalamus is not just a relay station, but it also estimates all this information and it synchronizes the cortical information during cognition. So it fine-tunes and weighs all the information. The drawings of Louis are much nicer than the MR images because on histology you can see the different thalamic nuclei, so they have also been drawn by Louis, but they're not visible on imaging. The different thalamic nuclei are important because they have different functions. In the thalamus there's this Y-shaped white matter structure that's called the internal medullary lamina. And then there's the anterior thalamic nucleus receiving information from the mammillothalamic tract and projecting to the cingulate. So this is a very limbic structure. The lateral group has connections with the motor cortex and reciprocal with the basal ganglia. The medial group has a function in the sleep-wake cycle and close connections to the hypothalamus. And then on the posterior side, there's the pulvinar that sticks out a little bit. And the word pulvinar means cushioned seat, that sticks out a little bit. And there is a restaurant in Tarragona that I visited for a neuroradiology course when I was still in training in 2008. And we like to go there with the neuroradiologists and the neuroradiologists in training, because it was also sticking out on the square like the pulvinar was sticking out of the thalamus. And on top of the pulvinar are the lateral and medial geniculate bodies for visual and acoustic information. There are two things that the thalamus can do in epilepsy. If there's a cortical focus with an epileptogenic network, the thalamus can play a role in the network because it synchronizes and weighs all this information. And it has been shown already more than 20 years ago that there's thalamic atrophy ipsilateral to the seizure focus in patients with temporal lobe epilepsy, and the amount of atrophy increases with the duration of epilepsy. So this is a secondary consequence of the epilepsy and of the seizures. Sometimes there's no cortical seizure focus, but a subcortical origin. And one of the possibilities for a subcortical origin might be the thalamus. The thalamus plays a role in the epileptogenic network. And there was this hypothesis that if you have focal seizures or if you have focal seizures with secondary generalization, the secondary generalization is mediated by the thalamus. And there was this nice multi-scale study where they looked at neuropsychological testing and fMRI studies in 130 patients with temporal lobe epilepsy, and indeed they found that if there was secondary generalization, there were abnormal thalamocortical connections. Sometimes you can also see differences in signal intensity in the thalamus. This is quite rare. This is a single center study where they collected patients over many years, and of the 5,500 patients, only 43 had a thalamic lesion, so that's less than 1%. And these thalamic lesions could be categorized into pulvinar lesions similar to the ones in status epilepticus, kystic lesions, bilateral hyperintense thalami in a metabolic disease called PALG that we will discuss later, and a linear defect in the anterior nucleus of the thalamus that, as I mentioned, is connected to the limbic system, and that was associated with atrophy of the limbic structure. So if you see an anterior linear defect in the thalamus, check the fornix, mammillary bodies, and hippocampus. There have been attempts to parcellate the different thalamic nuclei on MRI with DTI or by projecting histological data over T1 images using a probabilistic atlas, and then sometimes also DTI is included, but the DTI is mainly used to delineate the corticospinal tract and not so much for the intrinsic thalamic connections. Sometimes patients are drug resistant, and then neuromodulation might be an option to terminate the seizure propagation. There are 4 nuclei in the thalamus that have been proposed, and only the anterior nucleus is FDA approved. From an anatomical point of view, I think that the fact that there is white matter isolating the anterior nucleus contributes to the success of stimulation of the anterior nucleus, because if you would stimulate the pulvinar, you also stimulate the lateral nuclei. And even if you stimulate the anterior nucleus, there can be psychiatric side effects, as you can see in this case. So the anterior nucleus has been targeted and the electrodes have been inserted, but they had to discontinuate because of the side effects. And to conclude, I want to refer to a case that was published in Neurology this year that was very impressive. And it narrates of a female patient with right frontal lobe epilepsy with secondary generalization and very rapid spread. So the hypothesis was that there was an extensive network, and they were more for neuromodulation than for resection of the frontal focus. And to confirm this hypothesis, they implanted stereotactic EEG electrodes, and during this implantation, the patient had an electrographic status epilepticus, and they terminated it by acute thalamic stimulation. I've also pasted the EEG findings here, but I'm not so familiar with it, so please look up the article if interested. And with that, I think before continuing with hypothalamic hamartoma, I'll first spend a few minutes on the embryology of the deep structures of the forebrain, so thalamus, hypothalamus, and also a little bit caudate and hippocampus.